Dear viewers, today we see hormone assays, bioassays and immunoassays, RIA and ELISA, immunodiagnostics for pregnancy, cancer and reproductive tract infections. The topic has been divided in two parts. So, hereby in first part, we will be learning hormone assays, bioassays and immunoassays, RIA and ELISA. Introduction. Bioassay, commonly used shorthand for biological assay, is a type of scientific experiment. A bioassay involves the use of live animal or plant, that is in vivo, or tissue or cell, that is in vitro, to determine the biological activity of a substance such as a hormone or drug. Bioassays are typically conducted to measure the effects of a substance on a living organism and are essential in the development of new drugs and in monitoring environmental pollutants. Both are procedures by which the potency or the nature of a substance is estimated by studying its effects on living matter. A bioassay can also be used to determine the concentration of a particular constitution of a mixture that may cause harmful effects on organisms or the environment. Immunoassay is an analytical technique useful for the separation, detection and quantitation of both organic and inorganic analytes in diverse environmental and waste matrices. Immunodiagnostics is a diagnostic methodology that uses an antigen-antibody reaction as their primary means of detection. Bioassay, the determination of the relative strength of a substance, for an example, a drug or hormone or toxicant, by comparing its effect on a test organism with that of a standard preparation, is called bioassay. Bioassays are procedures that can be determine the concentration or purity or biological activity of a substance such as vitamin, hormone or plant growth factor by measuring the effect on an organism, tissue, cells, enzyme or receptor. Bioassays may be qualitative or quantitative. Qualitative bioassays are used for assessing the physical effects of a substance that may not be quantified, such as seeds fail to germinate or develop abnormally deformity. An example of a qualitative bioassay includes Arnold Adolph Berthol's famous experiment on castrated chickens. This analysis found that by removing the testicles of a chicken, it would not develop into a rooster because the endocrine signals necessary for this process were not available. Quantitative bioassays involve estimation of the concentration or potency of a substance by measurement of the biological responses that is produces. Quantitative bioassays are typically analyzed using the methods of biostatistics. Purpose: measurement of the pharmacological activity of new or chemically undefined substances, investigation of the function of endogenous mediators, determination of the side effect profile, including the degree of drug toxicity, measurement of the concentration of known substances, that is, alternatives to the use of whole animals have made this use obsolete. Assessing the amount of pollutants being released by a particular source such as wastewater or urban runoff. Determining the specificity of certain enzymes to certain substrates. Types of bioassays. Bioassays are of two types. Quantile. A quantile assay involves an all or none response. For example, insulin-induced hypoglycemic convulsion reaction or the cardiac arrest caused by the digitalis. The response is either positive or negative. There is no intermediate response. For example, either convulsion occurs or does not occur. Similarly is with cardiac arrest. In case of toxicity studies, the animal receiving a dose of drug either dies or does not die. Also, no intermediate response is possible. This is also known as the all or none response assay. The quantile method, though not precise, is employed for bioassay of substance in the following ways. Comparison of threshold response or comparison of effective dose, that is ED50, or medial lethal dose, that is LD50. Graded assays are based on the observation that there is a proportionate increase in the observed response following an increase in the concentration or dose. The parameters employed in such bioassays are based on the nature of the effect the substrate is expected to produce. For example, contraction of smooth muscle preparation for assessing histamine or study of blood pressure response in case of adrenaline. Environmental bioassays. Environmental bioassays are generally a broad range survey of toxicity. A toxicity identification evaluation is conducted to determine what the relevant toxicants are. Although bioassays are beneficial in determining the biological activity within an organism, they can often be time consuming and laborious. Organism specific factors may result in data that is not applicable to others in that species. For this reason, other biological techniques are often employed, including radioimmunoassays. Water pollution control requires some industrial discharges and municipal sewage treatment plants to conduct bioassays. These procedures, called whole effluent toxicity tests, including acute toxicity test as well as chronic test methods. The methods involve exposing living aquatic organism to samples of wastewater. For example, the bioassay Ecotox uses the microalgae Euglena glaciaris to test the toxicity of water sample. Immunoassay. Immunoassays are bioanalytical methods in which the quantitation of the analyte depends on the reaction of an antigen and an antibody. Principally, these methods are based on a competitive binding reaction between a fixed amount of labeled form of an analyte and a variable amount of unlabeled sample analyte for a limited amount of binding sites on a highly specific anti-analyte antibody. When these immunoanalytical reagents are mixed and incubated, the analyte is bound to the antibody, forming an immune complex. This complex is separated from the unbound reagent fraction by physical or chemical separation technique. Analysis is achieved by measuring the label activity, for example, radiation, fluorescence or enzyme in either of the bound or free fraction. A standard curve which represents the measured signal as a function of the concentration of the unlabeled analyte in the sample is constructed. Unknown analyte concentration is determined from this calibration curve. Immunoassay methods have been widely used in many important areas of pharmaceutical analysis such as diagnosis of diseases, therapeutic drug monitoring, clinical pharmacokinetic and bioequivalence studies in drug discovery and pharmaceutical industries. Radioimmunoassay, that is RIA. Radioimmunoassay RIA is a very sensitive in vitro assay technique used to measure concentrations of antigens, for example, hormone levels in the blood by use of antibodies. The technique of radioimmunoassay has revolutionized research and clinical practice in many areas, for example, blood banking diagnosis of allergies, endocrinology. The technique was introduced in 1960 by Yellow and Burson as an assay for the concentration of insulin in plasma. It represents the first time that hormone levels in the blood could be detected by an in vitro assay. Principle: the technique is based on the ability of an unlabeled form of a substance to inhibit competitively the binding of a radioactively labeled substance by specific antibodies. Method: to perform a radioimmunoassay, a known quantity of an antigen is made radioactive, frequently by labeling it with gamma radioactive isotope of iodine attached to tyrosine, that is hot. This radiolabeled antigen is then mixed with a known amount of antibody for that antigen, and as a result, the two specifically bind to one another. Then, a sample of serum from a patient containing an unknown quantity of that same antigen is added. This causes the unlabeled or cold antigen from the serum to compete with the radiolabeled antigen, that is the hot, for antibody binding sites. As the concentration of cold antigen is increased, more of it binds to the antibody, displacing the radiolabeled variant and reducing the ratio of antibody bound radiolabeled antigen to free radiolabeled antigen. The bound antigens are then separated from the unbound ones, and the radioactivity of the free antigen remaining in the supernatant is measured using a gamma counter. After determining the ratio of a bound to free antigen in each unknown, the antigen concentrations can be read directly from the standard curve. Application of RIA. Radioimmunoassay is widely used because of its great sensitivity. RIA as a major clinical tool, it is used to assay plasma levels of most of our hormones like HCG as a test for pregnancy, LH, luteinizing hormone, determining the time of ovulation, and TSH, thyroid stimulating hormone, T3 and T4 for thyroid function. Screening donated blood for evidence of viral contamination by HIV-1, HIV-2, that is presence of anti-HIV antibodies. Hepatitis C that is for the presence of antibodies, and hepatitis B, that is for testing for both antibodies and a viral antigen. Detection of sexually transmitted diseases like HIV, syphilis and chlamydia. Detection of anti-DNA antibodies in systemic lupus erythematosus, that is SLE. Narcotics, that is drug detection, early cancer detection, measurement of growth hormone levels, tracking of the leukemia virus diagnosis, and treatment of peptic ulcers research with brain chemicals called neurotransmitters. The main drawbacks of radioimmunoassay are the expense and hazardous of preparing and handling the radioactive antigen. RIA is generally simpler to perform and more precise than a bioassay, although it generally takes several days to perform. For comparison, ELISA can usually be performed in a single day. Enzyme-linked immunosorbent assay, that is ELISA. The ELISA, enzyme-linked immunosorbent assay, also sometimes known as EIA, that is enzyme immunoassay, is a rapid test used for detecting and quantifying antibodies or antigens in specimen against viruses, bacteria and other materials. This method is used to detect or diagnose infectious, autoimmune and other diseases. ELISA is carried out on solid matrix in 96 well microtiter plates or strips, that is 12 wells or 8 wells each, made of polystyrene or commercially available combs, that is for rapid ELISA. The protein antigen is affixed to any of the above mentioned surfaces, and then a specific antibody is applied over the surface so that it can bind to the antigen. This antibody is linked to an enzyme, and in the final step a substrate or substrate specific for the enzyme is added that the enzyme can convert it to some detectable signal, most commonly a color change. Performing ELISA, like other antigen-antibody reactions, involves at least one antibody with specificity for a particular antigen. Depending upon whether we want to detect antibody or antigen, the type of ELISA will vary. For example, the antigen in a sample can be detected either by direct ELISA, sandwich ELISA or competitive ELISA, and antibody is usually detected by indirect ELISA. Principle: simply put, for ELISA, an antigen is adsorbed or fixed to the surface of a solid matrix, which may be a well or a microtitration plate or well strip. Beads or maybe a special type of paper made of nitrocellulose. Then a specific antibody is applied over the surface so it can bind to the antigen. This antibody is linked to an enzyme, and in the final step, a substrate containing the specific substrate for the enzyme is added. The subsequent reaction produces a detectable signal, a color change in the substrate. ELISA technology has advanced a lot to make this immunoassay more and more sensitive as well as specific, various advances have been made, and now we have different types of ELISAs. There are slight differences in the technology, though basically ELISA remains an antigen-antibody reaction performed as wet test using different reporters and signal systems. Types of ELISAs. ELISAs can be divided into the following categories based on the principle used. Number 1, indirect ELISA. Number 2, sandwich ELISA. Number 3, competitive ELISA. And number 4, antigen and antibody captured ELISA. Number 1, indirect ELISA. HIV antigens are attached covalently to the solid phase support, allowing corresponding or specific antibodies present in the specimen to bind, and these bound antibodies are subsequently detected by enzyme labeled anti-human immunoglobulin and specific substrate system. If the test specimen contained the antibody specific to the antigen fixed on the solid phase, color reaction will take place. The color reaction can be read with naked eye or with ELISA reader at the specific UV light using the special filters. The result is expressed as OD, that is optical density value. Now, number 2, sandwich ELISA. One of the most useful for the immunoassays is the 2-antibody sandwich ELISA. This assay is used to determine the antigen concentration in unknown samples. This ELISA is fast and accurate, and if a purified antigen standard is available, the assay can determine the absolute amount of antigen in an unknown sample. To utilize this assay, one antibody, that is the capture antibody, is purified and bound to a solid phase, typically attached to the bottom of a plate well. Specimen containing antigen is then added and allowed to complex with the bound antibody. Unbound products are then removed with a wash, and a labeled second antibody, that is the detection antibody, is allowed to bind to the antigen, thus completing the sandwich. The assay is then quantitated by measuring the amount of labeled second antibody bound to the matrix through the use of a colorimetric substrate. Now we discuss number 3, competitive ELISA. In this assay, the antibodies present in the specimen compete with the enzyme conjugated antibodies in the reagent for binding to the antigen on the solid phase. If the test specimen contains corresponding or specific antibodies, these will compete with the labeled antibodies in the reagent for binding to antigen, so that less or not labeled antibodies can attach to the solid phase. Hence, faint or no color is produced on addition of substrate if specimen contains antibodies against the antigen on the solid phase. Now, number 4, antigen and antibody captured ELISA. Antigen capture ELISA can be based on the principle of indirect or competitive ELISA, only difference being in the initial step of attaching antigen to the solid phase. A monoclonal antibody directed against an antigen is bound to the solid support. Next step is addition of antigen supplied as reagent in the test kit. This antigen is captured by the monoclonal antibody bound to the solid phase. Test specimen appropriately diluted is added next. Antibodies, if present in the specimen, bind to the antigen on solid support. Remaining principle is same as indirect ELISA. Only advantage of antigen capture ELISA is that it is more specific than indirect assay. Method of ELISA. Although many variants of ELISA have been developed and used in different situations, they all depend on the same basic elements: coating or capture, direct or indirect immobilization of antigens to the surface of polystyrene microplate wells, plate blocking, addition of irrelevant protein or other molecule to cover all unsaturated surface binding sites of the microplate wells, probing or detection, incubation with antigen specific antibodies that affinity binds to the antigens, signal measurement, detection of the signal generated via the direct or secondary tag on the specific antibody. Applications of ELISA. ELISA is a highly sensitive and specific test. Since the discovery of ELISA, many advances have been made to make this assay more and more sensitive and specific. ELISA is employed in medicine to detect and diagnose microbial infectious diseases, autoimmune diseases, detection of antigen in a given sample. ELISA can be applied to detect and measure antibody in serum against viruses, bacteria, parasites. ELISA has also been used in home pregnancy test, that is rapid ELISA. ELISA is used in food industry to detect potential food allergens such as milk, peanuts, walnuts, almonds and eggs. ELISA can also be used in toxicology as a rapid presumptive screen for certain classes of drugs. The ELISA was widely used in various areas such as immunology, biological pharmacy, diagnostic industry and so on. Let us summarize. At the end, to conclude, this lesson described bioassays and immunoassays. The determination of the relative strength of a substance by comparing its effect on a test organism with that of a standard preparation is called bioassay. There are two types of bioassays, quantile and graded. Immunoassays are bioanalytical methods in which the quantitation of the analyte depends on the reaction of an antigen, that is analyte, and an antibody. We have studied principle, types, method and applications of RIA and ELISA techniques. Thus, in this manner, hormone assays, that is bioassays and immunoassays like RIA and ELISA has been studied to measure biological components of our interest in a given sample. In part 2, we will be learning immunodiagnostics for pregnancy, cancer and reproductive tract infections.